Welcome to Genomics in Java, a great place where we share science stories and conversation that we hope you'll enjoy as much as what's in your favorite cup. This morning, we're talking with Dr. Neil Lamb, the Vice President of Educational Outreach again, and we're going to dive deeper into that story that we mentioned last time about wine and cheese. So I love this time of year as the leaves start changing and it gets to be time for apple picking. Fall has arrived. In wine country, for wine harvesters and wine connoisseurs alike, fall mentions harvest time and it brings all the things that come with that. How is genetics and biotechnology helping with the wine industry?
It's a great question. And you're right, you and I both have our Java beverages here, but we're going to talk a little bit about wine and cheese, which is definitely not a Java conversation. I'm just going to slide that to the back. So to answer your question, genomics is used a lot in the wine industry to help us understand how genetic changes in a particular style of grape impact its flavor profile, or actually help the grape to grow better under certain kinds of climate conditions. The story that's in this year's guidebook, though, is a little bit different. It's less about the genome of the grapevine and more about the genome of phylloxera, which is an aphid-like pest that is the bane of existence for wine vineyards everywhere. And so the challenge is that this aphid-like pest either attaches to the leaves or the root of the vine, and essentially, do you remember the Capri Sun package? You take that straw and you.
I do.
Yes.
Okay,
so the phylloxera essentially does that with the grapevine. It just plugs right in to the nutrient system. It kind of like sets up its own beverage bar.
It's like the guest that never leaves.
It is like the guest that never leaves. And in fact, it actually builds kind of a gall, that's the formal term, this kind of ball of cells around it. It somehow manipulates the plant's defenses. And this can be really problematic when it happens in the roots, because it puts holes in the roots that make the vine susceptible to other infections and ultimately can kill the vine. So the guidebook story this year is the sequencing of the genome of the phylloxera, of the pest. And the interesting thing about that is that they identified 2,700 genes that are uniquely found in this little creature that somehow create proteins that mimic the instructions of the grapevine.
So this sounds a little bit like science fiction. There are 2,700 genes in this insect that mimic things that are in the grapevine.
Yes, it's a really incredible system to try to get the grapevine to behave the way that the phylloxera wants it to. So this understanding potentially begins to open the door for how we think about ways to develop more resistant plants or ways to try to treat phylloxera infestations. Now, what's really interesting about this is that this aphid-like creature probably first developed or first arose in North America. So over thousands and thousands of years, the phylloxera and the North American grapevines lived together. We would say that they co-evolved. So each of them were kind of in this arms race, the phylloxera trying to get as much nutrients as possible without killing the plant, and the plant trying to figure out how to just allow the phylloxera to have its minimum amount of nutrients and survive. But over time, they evolved a set of kind of symbiotic relationships. In the 1860s, the phylloxera was unintentionally brought to Europe, and European plants had not had that benefit of that long-term relationship.
This did not go well for the European
It did not go well. In fact, it almost led to the end of French wines. So what vineyard owners figured out they could do is they could graft their European vines onto North American roots, because the North American roots gave them some resistance. Better understanding the genome of phylloxera may help us avoid that practice entirely not too far down the road.
That is fascinating. Thanks to the field of genomics, the next time that I enjoy a glass of wine, I will never think about it quite the same again.
Yeah.
So, one of the things that I enjoy doing is sitting down with friends and a glass of wine and cheese and having fantastic conversations. But one of the things that I've become more aware of is that not everyone eats cheese. And so how do we help our friends who have chosen to live mainly plant-based dietary food lifestyles to be able to join in on the festivities? Is there anything that Genomics and Biotech can do to help us with that?
Yeah, there actually is a lot about that. So you can walk into your favorite grocery store nowadays and you will see non-dairy cheese alternatives. I brought a couple of them with me. So we've got a cheddar style cheese here in orange. And it actually primarily comes from coconut oil and pea plant protein. And then we've got a Wensleydale style that is also primarily coconut. But you can find cheese alternatives, vegan cheese made from nuts or made from other things. The challenge is they are not identical dairy proteins. So they don't have the same flavor profile. They don't melt in the same way. So some biotech companies are now creating dairy identical proteins. And dairy identical proteins are proteins like casein, for example, which is an important protein found in cow, sheep, goat milk that is an important part of cheese making. They've taken the genetic recipe from the cow, goat, or sheep, and they've inserted that recipe into a microbe, maybe a yeast or a bacteria. And so it reads that recipe and it creates the exact same dairy identical protein, but there's no cow involved.
So is this something that happens typically in biotechnology where we're able to take a genetic recipe from one organism and get another organism to produce it?
Yes, it is. Genetic recipes, the beauty of DNA is all living things use it. So any organism given a piece of DNA knows what to do and how to build the instructions, the protein from that. So for example, we get insulin from, we've taken the human insulin gene and we've put it into a microbe, and it now produces large amounts of that protein.
And so this cheese, this dairy identical protein, is produced in much the same way, but now we're producing the casein protein instead of insulin.
That's right. And then you blend it with other plant-based fats or other flavor profiles, and then you get curds, and you pack it and you age it just like you would any other cheese.
That is fascinating. I've always enjoyed looking at the cheese section at the grocery store, and so now the next time I'm going to have to pay a little bit more attention to those vegan cheeses. Thank you so much for sharing that information with us, and until next time, we hope that you'll continue to enjoy these conversations as much as you enjoy what's in your favorite cup.